Okay, so lecture got cut off a little bit early yesterday. The camera shut off a little bit early, so I wanted to finish out chapter 11 for you guys before I post the quiz today. And we left off talking about mutations. So sometimes in the process of DNA replication, DNA polymerase III doesn't attach the exact correct complementary nucleotide to the template strand. And anytime there's a change in the DNA sequence, whether it's in the template or complementary strand, that is by definition a mutation. And those mutations occur in a couple of different ways. One is a point mutation that we'll talk about in just a few minutes, and that's where a single nucleotide amid the long sequence that is the gene is changed. There can also be something called a recombination event, and that's where portions of one non-homologous chromosome are transferred over onto another non-homologous chromosome. Recombination, by definition, is a chromosome that consists of DNA from multiple sources. Now, we actually take advantage of that with DNA technology. When we're creating genetically modified organisms, we intentionally splice regions of other organisms, their DNA, into the target species that we're trying to genetically modify. Anyways, within our own genomes, that can occur as well. In fact, we know that it occurs, right? When meiosis 1 is occurring and we have synapses during prophase 1, we get crossing over, and that exchanges equitable regions of homologous chromosomes. Recombinant mutations, however, are a little bit different. This is where perhaps inequitable portions of non-homologous chromosomes are exchanged, and it causes a bunch of problems we'll talk about in just a few minutes. So the source of mutations there are a variety of them. Firstly, there are replication mistakes, like we were just talking about. So when we're replicating our DNA during the S phase of the cell cycle in interphase, sometimes DNA polymerase III or polymerase I might make a mistake and attach the wrong DNA nucleotide. Secondly, there are things called mutagens. There are physical or chemical substances that can physically change our DNA. We know that tobacco, for example, tobacco products are mutagenic, and so lots of the cancers associated with smoking or chewing tobacco are a product of these chemicals that are interacting with our DNA in bad ways. There are also physical phenomenons like radiation. Ultraviolet radiation is a really good example of that, where we have these high energy photons that are interacting with our DNA and physically changing them. Luckily, our cells have like a proofreading system, and they're really, really good at identifying replication errors and changing them and fixing them. If they're irreparable, then we know that the cell irrevocably enters a G sub 0 phase until ultimately it's digested through apoptosis. Okay, so the kinds of mutations. First of all, regions where they can occur. If a mutation occurs in the germ cells, or excuse me, the germ tissues, that is where we're creating gametes, then it's possible that those mutations are passed from a parent to an offspring. Alternatively, if they occur in the somatic cells, then generally they stay within the individual in which that mutation occurred. So most cancers are a product of some mutation in somatic cells, things like brain cancer and breast cancer, pancreatic and colon, lung cancer. Those are all somatic issues where mutations have occurred within somatic tissues. So let's talk about these two different types of mutations, the first of which is a point mutation, and the second of which is called a frame shift mutation. So with a point mutation, that is along the long sequence that is a gene, there can be a single nucleotide that's switched from, let's say, like an A to a T. So just one nucleotide is changed. Now, that can be problematic because ultimately we know that it can change the amino acids that are attached within the protein that gene sequence is coding for. So sickle cell anemia is a really good example of that. Sickle cell anemia is a product of a point mutation, a single nucleotide within a long gene sequence that was changed. And as a consequence of that, an amino acid was changed within a single protein within red blood cells, in the beta hemoglobin protein. Now that single amino acid changed the shape of the beta hemoglobin and consequently the shape of the entire cell. And so point mutations can be pretty dangerous. So if this is some gene sequence that we have, it's AAG, TTA, CCG, TAT. The way that DNA is read and the way that we make proteins from that is in a 3-letter sequence that are called codons. You can think about a gene as like a sentence, and that sentence consists of words, and those words are what we call codons. Each codon consists of a 3-nucleotide sequence. And so in this gene, there are 4 codons, and the first codon is AAG, so that codes for some amino acid. The second codon is TTA, and that codes for another amino acid. And then CCG, and then TAT. With a point mutation, what happens is a single point in that gene sequence is changed. So what used to be a T got switched to a C. So the first codon isn't affected. That second codon, however, is affected. So that's going to code perhaps for a very different kind of amino acid. The third and the fourth codons remain the same. So with a point mutation, you get a single change within a gene sequence that changes perhaps a single amino acid. The other kind of mutation, which seem a little bit more insidious and dangerous, are frame shift mutations. That's where either a portion of the DNA is removed or a novel portion is inserted. And this commonly happens with recombination events. What really happens, the ultimate consequence of that is everything downstream from that mutation site is shifted a little bit. Let me tell you what I mean. So here is, again, our original sequence, and then here's a frame shift mutation where at this point in that second codon, instead of that 4th nucleotide being changed, rather a new nucleotide is inserted so that everything downstream from it moves to the right 1 spot. So the first codon isn't affected, right? Because the insertion mutation occurred after that first codon. So AAG, first codon here in this new gene sequence, and AAG in the original sequence. Now the second codon should consist of a TTA, but because there's been a new nucleotide inserted, it now consists of GTT. So that codon is different from that codon. And it's shifted everything downstream from it to the right. So all of the subsequent codons have changed a little bit. What should be in the third codon, CCG, is now ACC. And what should be in the fourth codon, TAT, is now GTA. And so frame shift mutations shift the reading frame of the cellular components that we use to construct proteins, either to the left or to the right a little bit. And everything downstream from that mutation, all the codons downstream from that mutation, are changed. And that causes all sorts of insidious problems. Okay, so that was a rather brief introduction to mutation. Please, if you have questions or concerns or comments, don't hesitate to call or email me.